Hi, everyone. Good afternoon. I am Maria Paula, member of the organizing committee of Unicamp's Chemistry Week. First, I would like to give a warning to the Chemistry Institute graduate students. By the end of the lecture, we will share through the chat a Google Form link to verify your participation, so stay tuned. This year's special guest lecture called What I've Learned from a Life in Science will be given by Dr. Bruce Alberts. Bruce has graduated in biochemical science at Harvard College, got his PhDs in biophysics at Harvard as well, and his postdoctoral at Geneva University. Worked in important universities and institutions such as Princeton University Science Magazine as editor-in-chief, the National Academy of Science, and one of the first 3 United States science envoys. Alberts is also noticed as one of the original authors of the book called The Molecular Biology of the Cell. He is now the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California, San Francisco. His current project involves supporting young scientists, improvement of science and mathematics education, empowerment of teachers, and redefinition of biochemistry teaching and research. I would like to inform you that Dr. Alberts will answer your questions by the end of the lecture. The organizing committee will be collecting the questions from the chat, so you may send them anytime during the lecture. I ask you, if possible, send your questions in English, but if you are not able to do so, we will try to translate them. Now, I will leave you with Dr. Bruce Alberts. I wish you all a great lecture.
Hello, can you hear me? Is this working?
Yes, it is.
Okay, very good. We had a little technical problems at the beginning. It's a real pleasure to be here remotely. I wish I could be there in person. I always enjoy coming to Brazil. The young people are so enthusiastic about everything, life, science, and my field of cell biology. So it's really great to interact with you. My major message is that there's a huge need for whatever you're going to do in science in the future. There are many things you could do, but the world needs much more science. And I'm going to try to explain that after I give a little history about myself and what I've learned mostly from failure. And the major other message of this talk will be that everybody fails, and those who are successful learn from their failures. And for that reason, older people who have failed a lot have something to offer to younger people in the way of advice. We become wiser from all our many failures. Okay, so this is the outline of my talk. I'm going to talk for 90 minutes, which is longer than I ever talk, but I'm not sure I could last that long. But anyway, so I have a lot of things to cover. I'm going to talk first about my life in science, the biochemistry of protein machines, which we had no idea that proteins form machines when I started. But that's really the basic of the chemistry of life depends on thousands of protein machines. I'm going to talk about then writing textbooks, which I've done a lot of. I'm still working on that. 43 years of writing textbooks. Then science policy by accident. I'll explain how I got to Washington, D.C. At the National Academy of Sciences, and then after that at Science Magazine. But most importantly, what did I learn? Why is science so critical for the world? I think that science is much more important for the world than most scientists understand or think. And I only learned that from all my many experiences mostly in these other jobs besides doing science. And then I'm going to emphasize my major passion now, the urgent need for reinvigorating science education in new ways, making it focus on a different outcome than the outcomes we often seek in science class. I'll explain that later. Okay, start my life in science. So I was a high school student near Chicago, Illinois, in the Middle West of the United States, when this paper appeared by Watson and Crick in 1953. Of course, as a high school student, I knew nothing about it. I only learned about it a little bit later. But the important thing I want to say about this picture is that Jim Watson was only 25 years old, and we need to encourage young scientists to be ambitious and give them a chance to contribute, because that's when you have your most energy and your most original insights. So Watson and Crick had really solved suddenly the real mystery of heredity. How could you store information in such a small space and have it remain stable and be replicated for millions of generations of cells? That in 1952, people had puzzled about that, and they thought about it much like we think today about the human consciousness. How can we explain human consciousness? It comes from a set of nerves in our brain. It seems like something impossible to explain. And before the Watson and Crick paper, people struggled with heredity in much the same way. But their structure of DNA just immediately changed everything in that regard. And it was incredibly exciting for my generation. So they didn't know any chemistry. Actually, neither one knew any chemistry, Watson or Crick. Crick was a physicist, Watson was a biologist. And they didn't know anything about the machinery that enabled a DNA sequence to be copied from one generation to the next. And so starting as a graduate student in 1961, I spent more than 30 years working on this problem. So shortly after Watson and Crick, 4 years later, Arthur Kornberg, in 1957, made a very exciting discovery that earned him a Nobel Prize. He discovered an enzyme called DNA polymerase, which is the enzyme that catalyzes the addition of one nucleotide to the 3' end of a primer strand, as illustrated here. And what Kornberg showed is that which of the 4 nucleotides is added is selected by matching the template strand, and that's how information is passed, as Watson and Crick had predicted. And at that time, I was in a chemistry lab, actually, doing my PhD, and we thought that the cell was just like a test tube. It was just full of a random mixture of very concentrated proteins and other molecules, and everything was just randomly collided very rapidly with every other molecule, because we knew as physical chemists, I was in a physical chemistry lab, that the rates of collision are very, very rapid. And so we thought that everything would just work by random encounters of an enzyme with its site of action, and this is DNA polymerase, would then bind to the primer strand, add one nucleotide, fall off. The next DNA polymerase molecule would bind, add another one, and so on. Just everything would happen in this random mix. And of course, this turns out to be wrong, and I'll explain that. But this is where we started, and it's really astounding. And scientists are optimistic. We've seen so much progress from science over the course of our lives, and so that optimism about solving problems is important for humanity, and in many different ways, as I'll explain. So I am now going to introduce my first of many failures. So I had a very dismal career as a graduate student getting my PhD at Harvard. I had been inspired by the success of Watson and Crick, and therefore I made my own theory about how DNA polymerase, just one enzyme, might be able to catalyze DNA synthesis on a chromosome. One of the problems was that DNA polymerase in the test tube only worked on single-stranded DNA, and of course DNA is a double helix. So I did many experiments trying to see if my theory was right, and of course my theory was not right, and the no answers did not surprise anyone. They did not add to the store of scientific knowledge. Nevertheless, my professor, after 5 years, said I'd done enough experiments. He helped me write my thesis. I wrote a long thesis, and I submitted it to my committee. And everybody at Harvard had to sit down with a group of 4 or 5 professors and talk about what they did for their PhD, and then this committee would automatically pass you. That's what I thought. Nobody had failed in the recent years, but that didn't stop them from failing me. So I was shocked to discover that I would have to spend another 6 months earning my PhD, because they didn't think I had done anything important scientifically. They didn't believe what I had found. And that failure, as I've explained in this published piece in Nature in 2004, really caused me to think carefully about how to do science and study my mistakes. And what I learned, I think, was the basis for my success as a scientist later. Basically, I think we learn from our failures. It's very hard to learn from your successes. I had been pretty successful as an undergraduate doing research. I was very lucky, but I didn't learn how to do science. So what did I learn? The first thing I learned was that theoretical biology is much more difficult than my generation had imagined. We had been misled by the striking success of the 1953 Watson-Crick DNA model. And really, having a good strategy in scientific research is the key to success. I sometimes talk about the fact that if you ask me to paint a painting, you could give me all of the paintings and materials that Rembrandt had, a great Dutch artist. You give me all his materials, and yet I couldn't make anything beautiful out of it. The same thing is true of science. Science is a creative endeavor. It's not just knowing how to do different techniques, it's how you use them. The strategy with which you design experiments and research projects, that's what's the creative part of science. And just like art, good science is very creative. So finally, they did pass me after 6 more months of doing work in the lab, rewriting my thesis. And then I was able to go on to my postdoc. In 1965, I went to Geneva, and I worked with these 2 individuals, and suddenly I discovered that my whole idea for my thesis was wrong, because I had thought that DNA replication required only one enzyme, but in fact, I discovered that genetics done on this model organism, virus, bacteriophage T4, had genetic analysis had done an extensive analysis showing that there were at least 7 genes that were needed for the DNA of the virus to replicate. And so basically, there are these 7 proteins needed for DNA replication, not just 1. And this paper I hadn't seen. So learning from failure number 2, read very widely in the scientific literature. I had been only reading biochemical papers, not papers in genetics. And like most biochemists, I had thought that only 1 protein was needed to replicate DNA inside the cell. Okay, so now I knew I needed strategy, right? I had failed already, so I needed a strategy that was different from what other people had. And I made an investment during my postdoc of developing a new method, DNA affinity chromatography. It turned out I could stick DNA onto plain DNA cellulose column and make a column out of DNA. And many proteins that function on DNA in the cell will have a site on their surface for binding to the DNA. We already knew that. We knew that that binding would be sensitive to salt because there's electrostatic component, important electrostatic component of any proteins binding to DNA, because DNA is highly negatively charged. So proteins use positive charge on their surface to help their DNA binding. So I could, by fixing a high concentration of single-stranded or double-stranded DNA on a solid support, one should be able to trap these proteins specifically and then elute them with salt and purify them. And I found that already in my postdoc that many proteins in the T4 bacteriophage infected cell bound to DNA cell's column. Would one of them be missing if a mutant T4 virus was used for the infection? And particularly, I was interested in the mutants that failed to make DNA inside the cell. So after a one-year postdoc in Geneva, I started my own small laboratory as an assistant professor at Princeton University in 1966. I had undergraduate and graduate students, a very small lab. The students and I focused on purifying each of the 7 bacteriophage T4 proteins identified by the 7 mutant genes. And our hope was that someday we could mix those 7 proteins together in a test tube with double-stranded DNA and get the DNA to replicate. Well, I had a great, finally did happen, took 9 years, finally happened in 1975. This is a picture of my Princeton lab. We had a lot of fun. We were all young. I don't know if you could recognize which one is me. I'm not the dog, but I'm the guy in the back with the red shirt and the white suspenders. The guy with glasses looking with a tie, that's Keith Yamamoto, one of my first graduate students. He was very successful. He's now my boss at UCSF. He's some kind of vice chancellor. So this is the way the world works. So I'm very pleased that many of these students went on to have successful careers. So what we discovered was a protein machine. It's a complex of 7 proteins that makes a new DNA helix by copying an old one, just as predicted by Watson and Crick. This requires that 2 DNA polymerases work at the same time. And because the 2 strands of the DNA helix run in opposite directions, they're anti-parallel, 2 polymerases one of the polymerase has to work backwards on the lagging strand, making DNA as a series of short pieces called Okazaki pieces. And the whole structure is folded over, much like shown here, creating a loop to enable the lagging strand to function. I have the magic of protein machines is best appreciated by a movie that shows a machine in action. I'm not sure this movie is going to work, but we'll see what if it doesn't. I'll try to explain. Okay, here. This is a movie that was made at the 50th anniversary of the discovery of the DNA double helix. And it's really a wonderful way of understanding how protein machines work in general, but they all work differently, of course. Let's see if it works. It may not work. Oh, sorry. This happens often for some magical reason. When I'm doing a talk, this won't work.
Dr. Bruce, try on the bottom of your screen if there is any button so you can play it.
Yeah, I don't see any button. It sometimes works and sometimes doesn't. I
Try
will,
to click on the image.
yeah, I will. I'm clicking on the image and I'll try one
It's
more.
not working. Sorry.
Okay, no problem. Anyway, I will send a link to the movie to the organizers in case you want to see it. Okay, but it's really amazing how biology works through these very sophisticated coordinated movements of proteins. Uh, and this requires, just like the machines in your, that you use every day, require electricity to make directional movement. These machines require the hydrolysis of ATP or GTP to drive allosteric protein movements. This is amazing chemistry. I know you're all chemists, but the chemistry of life is by far the most sophisticated chemistry known. Okay, 2 lessons learned in the last 30 years. First of all, because of evolution, there are remarkable homologies between living things. Therefore, use model organisms wherever possible to start to work out mechanisms. I worked on this T4 bacteriophage. Other people worked with E. coli. Those systems are less complex. They have the same mechanisms, but they don't require as many proteins as human replication does, because our replication is much more highly regulated. So instead of 7 proteins, we might have 20, but the basic mechanism is the same. So the shortcut to understanding humans very, very often is understanding how a simpler organism does the same kind of process. Secondly, nearly all cell processes will be driven by 10 to 20 proteins organized as a protein machine and incorporating ordered protein movements driven by the energy of ATP hydrolysis. And finally, they'll be based on elegant mechanisms that are too complex to predict. I was trying to predict how DNA replicated as a graduate student. There's just no way of you had to discover it. If they had told us it would be this complicated when we started, we wouldn't have believed anybody. All right. Now on to my next near failure. So in 1978, I just moved from Princeton to the University of California to San Francisco, big medical school where I am now. I was sitting in my office. I still remember that I get a phone call from the famous Jim Watson. He's now 50 years old. He's no longer 25 years old. And he's quite famous, of course. And Jim Watson was always very creative and insightful. And he had the very correct idea that this was the time to unite 2 different fields. One was molecular biology, the kind of work that I did, trying to figure out, starting from chemistry, how cells worked. And the kind of work I did on DNA replication, of course, was being done in many other areas, protein synthesis. We understood the genetic code. All those things came out of molecular biology, really based on chemistry. But there was a whole other field at that time called cell biology that came out of originally the detailed light microscopy in the 19th century, looking carefully at cells and naming their parts. And trying to figure out how things worked. And of course, that field was very successful in discovering that chromosomes, for example, carry the hereditary material. It made many discoveries, and then electron microscopy in the 20th century added to that, made it more sophisticated, but it was almost completely divorced from molecules. And it was a field that in 1978, I didn't even understand it. I had tried to figure out what cell biology was, but it didn't make any sense to me. But Jim Watson said that this is a time to try to write a textbook to bring those two things together, to try to bring a molecular basis to all the observations that cell biologists had made. And he always gets people to do things by having great ideas and totally underestimating how much time it would take. He said it would only take us 2 summers, the summer of 1978, and then we would finish it in the summer of 1979. So in the summer of 1979, we went to a house near Cold Spring Harbor, and all the authors sat there for 2 months trying to finish the book, and we realized that this was not going to happen. This is a picture from this house. There's an old house that Jim Watson rented. That's Jim Watson in the white shirt, and next to him with all that hair facing the camera is me. But here we are discussing, how we're going to finish this textbook in just another month. And this is my close colleague, a wonderful friend now, Martin Raff. We just realized that this is going to be enormously harder than we had thought and take much more time. This is what textbook writing looked like before laptop computers. We do a first draft, type it out. Somebody else would then go over the type copy. Somebody else would go over that. I think the writing in pen is mine. This is another author's textbook. I mean, the reason why our textbook actually turned out well is that we all worked on the chapters. Not many textbooks, each chapter is written by a different person. But this is what it was like, and the Martin Raff's wife did much of the typing because she was a great typist. And it just was a enormous chore, but writing took so much time. So finally, I think I call it the miracle of 1981. We worked Christmas and summers, and suddenly in 1981 we started to be able to write chapters that made sense, and eventually we finally published the first edition. Producing this book was very much harder than we had expected. My colleague kept track of how many days we spent. We spent over a year of 12-hour days writing this textbook. Here's the starting set of authors crossing Abbey Road. We took this picture because we were working in, all the other authors were except me and Jim Watson were from UK. So we did most of our writing in London, and right around the corner from the Abbey Road recording studio. And this picture was taken as a joke, but then it ended up being on the imitation beetle covers on the back of our third edition, and that became a feature of all our textbooks, putting crazy imitation beetle covers on the back. This is the 7th edition cover. It's going to be, because of the pandemic, this is the first edition we've actually finished on time, and it'll be published in February 2022. I just want to make the important point that each time we write a new edition, we are humbled by how much we still don't know. That's great for all you young students. There's so much more to discover. We need you to do the discovering. I just want to give some examples of 2 recent surprises for textbook authors, of many I could list. The first is the recognition that the extensive set of scaffolds, special protein and RNA molecules, produce biochemical subcompartments in the cell without requiring a membrane, and that many of these scaffolds can assemble to create larger phase-separated liquid compartments. And the second one is the recognition that the chemistry is so complicated that it gives rise to what physicists originally called emergent properties. And I'll explain that later. Scaffold proteins. Here's a diagram from our textbook. It's simple chemistry. Obviously, if you have a reaction, as many reactions or chemical reactions are in biology, that require many different proteins to come together, you could speed them up by increasing the concentration of those proteins. And the way we know it's been done many times was put them inside a special membrane, like inside the nucleus or inside the lysosome and so on. And when we started writing the first few editions, we thought that was the only way you could concentrate proteins and speed reactions. But in fact, the most prominent one is to use scaffolds, which are protein or RNA molecules that have lots of flexible parts, but also bind all the different molecules that are supposed to react. So by doing this, they greatly speed the collision rate and the probability of reaction, and therefore are efficient ways of improving the chemistry and making it work better for cells. Here's a simple example. This is the polymerization of a important cytoskeletal protein in all of our cells. They're called actin filaments. These filaments are these red things that would go down all the way through the floor for many stories. They're very long filaments. This is the growing end. And the growing end grows at rates that are faster than diffusion controlled. That was a puzzle at first, but then it was discovered that the green protein that catalyzes the reaction of putting new actin filaments on the end and causing the filament to form and grow fast, that has all these scaffolds or whiskers that hold the actin monomers, the precursors, close by, so that the rates can be much faster than they otherwise would be. That's just a simple example. Here, RNA scaffolds are also widely used in cells. Those were more recently discovered. Many of them are so-called long non-coding RNAs or link RNAs. We don't know that much about most of them. Most of them still are not well studied. So scaffolds are involved in forming different biochemical factories inside the cell, which we now recognize are as phase-separated biomolecular condensates. Here's just a picture of a cell nucleus where RNA is being made in the red areas and DNA is being made in the green areas, and those so-called factories form only when they're needed, and then they dissolve and go away. So these are temporary condensates that speed either the synthesis of RNA, transcription, or DNA replication in green. Biomolecular condensates are now known to help to form many different so-called membraneless organelles. The most famous of and the one that was studied even in the 19th century is the nucleolus. Nobody understood how it came together. In early editions of our book, it was a real puzzle. How did we get this structure with no membrane around it? Now it's known to be a biomolecular condensate. The centrosome, synaptic densities, and so on. Still, a lot of work remains to be done on these relatively newly discovered features of cells. In conclusion, therefore, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds. These scaffolds both speed reactions between specific components and locate those reactions in a particular part of the cell, as I just illustrated in the last examples. Okay, so that's just a quick overview of one thing we had no idea about when we started writing this textbook. Emergent properties are something that really underlie all of cell chemistry because of the complexity. So here's nearly how nearly all of biology works. You know, you go A goes to B, goes to C, goes to D, goes to E, all catalyzed by enzymes. Or but basically those things would not work inside of a cell without careful controls. So there's feedback and feed forward loops that make these reactions actually useful for the cell. And there's no way to understand such pathways really without mathematics. And this is just a relatively simple one that get much more complicated than this. Here's a specific example, the great complexity of the actin filament network. Actin is a protein I showed you growing with the scaffolds in a previous slide. So there's a filament in the middle. Actin filament is actually much, much longer than that. And what actually that filament does and how it contributes to the cell's life depends on its interaction with some of these other proteins. And there's more than 50 of them in a yeast cell, but there's even more in a human cell. And some of them are like the protein I showed controlling filament dynamics at the end of the filament. Those are those dark purple ones. Others are motor proteins that move things along filaments. Those are the green ones. There are all these other proteins anyway, and they compete with each other in mutually exclusive complexes. And what actually happens to the actin filament depends on all these interactions, and in different parts of the cell, the actin is doing different things. And we really, even though we know all the components and we can measure all the rate constants, we don't have the methodology yet to be able to predict what happens inside of a cell. So as a consequence of such complexity, even when we gain a complete knowledge of all the molecules, protein machines, and molecular interactions in a cell, we will not be able to understand even the simplest of living cells. Instead, life reflects the emergent properties that result from very complex networks of chemical reactions. So, my conclusion, it will probably take at least the rest of the century to gain a true understanding of how cells and organisms work. Much more biochemistry will be needed in purified systems that reconstitute biological systems, all the protein machines, see how they actually work, not just what they are, but how do they work. We also need new quantitative methods for analyzing and understanding the enormous complexity of life chemistry. That's going to take computer modeling and mathematics, new methods that we don't yet have. So the current problem, for somebody who writes a textbook, I mean, we have a privilege. We every 5 years, we very carefully examine what is known, what's new, what isn't known. But most people don't have that privilege, so they don't realize how much is unknown. And so a current problem is that, at least in the United States, our main funding agency for biological research and has been overemphasizing translational medical research. Basically, work that's obviously related to some disease is going to, that's what we mean by translational biomedical research. But with so many unknowns and so little understanding, fundamental research on biological mechanisms also remains absolutely critical for improving human health over the longer run. It doesn't give you immediate results, but over the longer run, that's what's going to take to really help humanity deal better with health and disease. This is my favorite editorial. When I was at Science Magazine for 5 years, I had the privilege of organizing an editorial every week. And so there were like 250 editorials during my 5 years at Science Magazine. This is my favorite one by Huda Zogby, She's a very distinguished MD who worked with children having brain diseases, genetic diseases, and realized she couldn't solve them as a clinician. She then went into research, and she's been enormously effective as a translational researcher working on trying to cure disease. But she says the challenge in translational medicine is that scientists are trying to translate the text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience. And as I've outlined in yellow here, she regrets that so many highly qualified basic scientists today feel compelled to jump on the translational medicine bandwagon, because that's where most of the funding is available from the National Institutes of Health. Here's an example of how little we know. I talked about cells being the most what I feel they're the most amazing thing on Earth. They're self-replicating chemical systems, and here's the simplest minimal cell we know so far. This pioneering work got rid of all the genes in a small bacterium that were possible to get rid of, and they were left with 473 genes in what they called a minimal genome to make a self-replicating cell. And to everyone's surprise, including theirs, 149 of the 473 genes were of unknown function, and most of those are still of unknown function. This is 5 years later, even 5 years later. Another example is the importance of the fruit fly Drosophila. We're still far from understanding how cells work together to form and maintain tissues. It's just amazing chemistry. But many examples show that the first working out a mechanism in the fruit fly provides a shortcut to understanding humans. For example, in our last edition, the chapter on the development of tissues contains 50 references to the fruit fly, 4 times more than the next most cited organism, which is the mouse. So many things have been discovered in fruit flies, so many genes and so many processes, and then we recognize how to work on mice and humans. Consider the human brain. If you don't understand what emergent property means, just think of human consciousness. It's an emergent property that comes out of the connections of all those nerve cells in our brain. So this is from our textbook. Basically, the human brain contains more than 100 billion neurons, each of which on average has to make connections with 1,000 other neurons according to a regular and predictable wiring plan. It's incredibly complicated. The Drosophil brain contains about 100,000 neurons, 1 million times less than humans. We could map all the neurons. We could do genetics, knock out any protein you want and see what happens to the brain and its function. All these things we can't do with humans. And obviously working out how the Drosophil brain works will be a real shortcut to understanding our own brain. But the amount of resources put in this area is so much smaller than it should be, because it's not obvious how this is going to help cure disease, but it, of course, will over the long term. To understand cells, we also need new model organisms. This is a paper from my young professor at UCSF, Wallace Marsh, who had the courage to go back and work on a really old mystery organism. This is a giant single cell, the ciliate, called stentor, which have been studied extensively for about 100 years, and then everybody stopped working on it because they didn't know how to figure it out. But now we have new tools. Basically, this single cell has an intricately patterned plasma membrane it knows where to put a mouth. We cut it in little pieces, it regenerates itself. I would bet that human cells also have some kind of a way of determining where on the cell you are, but this is a cell that makes it really obvious, and it's obvious what we learn here will have certainly implications for humans over the long term. A new model organism, unfortunately, is the coronavirus, this disaster that has really affected the world and is still ravaging so many countries, such a tragic event. This virus is an unusually large RNA virus, more than 4 times larger than flu. It has 29 proteins, and most of those proteins we do not understand. The massive mobilization of scientists around the globe now focus on understanding the fundamental cell biology of this single virus is unprecedented, and it will lead to many new fundamental understandings of critical importance to the future of human health. You know, how are so many of the proteins of these large viruses are designed to interfere with our immune system, and they represent probes of how to help us understand how our immune system works, which again is something like the human brain we don't understand well enough, and this is going to help us understand it. So that's my bit on science, on the chemistry of life. I'd now like to go to my next episode of my life, closing my lab and moving to Washington, D.C. Okay, how did that start? Well, it's a complete accident. In 1985, I wrote this editorial, appeared in Cell, called Limits to Growth in Biology, Small Science is Good Science. And basically, I had been seeing the Howard Hughes Medical Institute put a lot of money into laboratories. They'd find really excellent scientists, and they say, we want you to double the size of your laboratory. So laboratories that have been, say, 15 people will now be 30 people because Howard Hughes was giving them all this money. And what I was observing is that generally it took excellent science and made their science more boring. So I wrote about why small labs are important. This article was then attacked by many other scientists in other issues of Cell that came later, arguing how I was wrong. One of them, I remember, one of my arguments was that in order to be able to be a good mentor to other young scientists in your lab, you have to do experiments sometimes so you understand the technology and the techniques. And so one of these articles said, if Bruce Alberts thinks that he should be able to do experiments, he should be paid like a technician. I don't know what. So anyway, I was famous for being, or infamous for not liking big labs, promoting small science. And that's the reason why I got this phone call a year later. I got a call suddenly in my office, the same place. I still remember where I got the call from Jim Watson. I was sitting in my office in San Francisco. Should there be a special project in the U.S. to map and sequence the human genome? A very prestigious committee had just been established by the National Academy of Sciences to address this question. It was full of famous people who had spoken out strongly on both sides of the argument. Jim Watson was all in favor of it. David Botstein and Shirley Tillman were all strongly against it. The whole thing was set up, and they needed a chair for the committee. Since I had not even thought about the question, they said I would be the ideal chair for this important committee. Actually, I think they meant I would be the ideal chair because I was not only unbiased, because I hadn't committed, I hadn't had any time to even think about it, but the biological community was strongly against this idea because it would make big science out of biology. Like they would say, they convert biology to big science like physics. And so therefore, if I was chair of this committee and we came out to favor a program, it would help make the biological community believe in the report, because I was a small science advocate. Well, why was any special genome so controversial, genome project so controversial in 1986? Why were most biological scientists against it? First of all, with available technology, our genome of 3 billion nucleotides at that time would require 30,000 person years to sequence. It was even suggested that it be done in prisons, making use of prisoners. The idea was also, as I implied, broadly viewed as a big science threat to the successful small science culture and small science funding in biology. So we produced this report, Mapping and Sequencing the Human Genome. All these people who were on different sides of the issue came to the consensus. And this is turned out to be one of the most successful reports that the Academy has produced. We produced many, many reports, as I'll explain. And I was thought to then be a good person to work on science policy, which I had never done before in my life. That led to another fateful phone call in late 1992. This was a special nominating committee to select the next president of the National Academy of Sciences had spent a year discussing who should be the next president. They had selected me for this full-time job in Washington, D.C. I had decided not to be a candidate when they had phoned me 9 months earlier, because it's a full-time job and I wanted to stay in my lab. I didn't want to continue to do what I was doing. Well, they said, well, they recognize us, but they put so much work. Please have the courtesy of meeting with the committee to discuss the possibility. Anyway, they convinced me to take this job, and my life then, I had to close my lab and suddenly shift from science to science policy. And I received it. All life is an education. And I received a major education in Washington, D.C. from 1993 to 2005, when I was full-time president living in Washington, D.C., as working on science policy. The U.S. National Academy of Sciences is more than an honorary society, because Abraham Lincoln gave us this special charter. The Academy shall, whenever called upon by any department of the government, investigate, examine, and report upon any subject of science or art. Art meant technology. Any subject of science or technology. And here's the catch. But the Academy shall receive no compensation whatsoever for any services to the government of the United States. Well, that made it impossible, really, to do the work that was required by the government. And in the first 10 years, the Academy almost died, from 1863 to 1873. But eventually they worked out a compromise where everybody who did studies for the government would be unpaid. Everybody would be volunteered, but the government would provide some money to run the Academy. And today, this is an incredibly important entity for producing the science to underlie policymaking. And with the advent of the World Wide Web, actually, when I came to Washington in 1993, there was no World Wide Web. 1994, suddenly we could make all our work available to the rest of the world using the web. And there are now more than 8,000 books online about everything you could imagine: arsenic in drinking water, climate change, all kinds of advice on everything you could imagine. And this science advice can be used anywhere around the world as free PDFs. So my first year in Washington was also nearly a failure. I inherited a staff of 1,000 people in a complex organizational arrangement. I knew almost nothing about Washington, D.C., or the government. I soon discovered that few of my decisions for new directions were being carried out. I mean, I didn't recognize a lot of the people who had been there for a long time didn't want to change, and they just pretended to change. And despite my working 120 hours a week, much seemed out of control. So some friends organized a secret meeting to see if they could help rescue me. It was that bad. That was about a year after I had been there. At any rate, finally, we made it work. I had to remove a lot of the lead staff, because they didn't want to change and so on. And I don't want to talk about most of what I did, but I want to talk about the most important part of my education at the Academy was the recognition of the critical importance of science for everyone. Here I am, and part of my Academy job took me to remote parts of Africa. And here I am with subsistence farmers. The woman with the bandana on is a subsistence farmer. The person with the tie is a Kenyan scientist. We're in a region where the Kenyan scientists had taught the farmers how to do research on how to grow their crops better. You could see their corn is now doing very well. These were villages that for thousands of years had been undergoing starvation during the non-growing period. They could never grow enough food to sustain themselves. But now the villages with this scientifically based agricultural research that they were doing in their own farms could produce so much food they were selling it to other villages. And of course, the technology is spreading everywhere. Here I am in India learning about how the wireless internet could be used to bring science to even the most poor farmers in India, with a huge amount of effort from my friend M.S. Swaminathan. I learned how the internet could help groups of Indian women in these villages produce biopesticides, prevent them from having to buy chemical pesticides, and enabling them to sell these parasitic wasp eggs in the marketplace to other villages. I also learned a lot in my 5 years at Science Magazine. This is the last issue that before I stepped down as editor-in-chief. And we had gone on a wonderful trip in April to India with Science News staff and discovered how all of these different advanced scientific institutes, the Indian Institute of Technologies and so on, were really bringing science to improve the village lives in many different areas. So another thing I learned early on, really, from my trips to India, was that every nation also needs much more of the creativity, rationality, openness, respect for evidence, and tolerance that are inherent to science, what Indian Prime Minister Nehru in 1950 called a scientific temper for the success of humanity. I had never heard the word scientific temper before, but now I use it all the time. We need people to have a scientific temper. The first Prime Minister of India, Nehru, recognized that his diverse country with so many different religions and languages needed the values of science in order to be a successful democracy. Here's some important values of science: honesty. Everybody has to be honest, otherwise science doesn't work. Generosity, and a strong demand for evidence with openness to all ideas and opinions, irrespective of their source. One has to recognize that a graduate student may have a better idea than a professor, otherwise science doesn't advance. So the central lessons from my 17 years in Washington, D.C., 12 at the Academy and 5 at Science Magazine were, first of all, it's critically important that science teachers and scientists achieve a much higher degree of influence throughout both their own nations and the world. And secondly, we need to refine what is meant by science education if we're going to produce a scientific temper across the globe. And I'm going to spend some much more time on this now toward the end of my talk. So what about creating a scientific temper for the world? This is not a new idea. The famous educationalist in the United States, John Dewey, wrote now 110 years ago, one of the only 2 articles that remain in my credo of life is that the future of our civilization depends upon the widening, spread, and deepening hold of the scientific habit of mind. And that the problem of problems in our education is therefore to discover how to mature and make effective this scientific habit. This is really going to be the focus of the rest of my talk. And what I'm trying to work on now, I've been, since I left Science Magazine, I've been working on that. So this issue seems even more urgent due to events in the past year. This will not be a surprise to anybody in Brazil. I'm talking about the US, but it applies much more widely. It is deeply discouraging that in the United States today, so many Americans feel comfortable denying what science knows on issues that range from protecting us from the COVID-19 virus to climate change. Today, in the United States, we have so much vaccine that nobody wants. They don't want it because they have some crazy idea that, they read this on the internet. There's microchips from Bill Gates in the vaccine. The vaccine is more dangerous than getting COVID. But it's all these crazy things that have no basis in evidence. And in that and other areas, there's so much evidence-free thinking. There's so many people who believe that our election was not fair, which it was. That they believe that the president wasn't really elected. This is a horrible, dangerous thinking for any nation. And I claim it represents a massive failure of science education. And I think we have an opportunity now, since all my colleagues who were teaching science in college now are aware of this danger, to really try to refocus science education to make it more effective in helping our democracies and our civilization survive. Science education is a critical tool for humanity, but the wrong way to teach science still predominates. Here's an actual example. This is true. I was told by a mother after I gave a talk at a university. Her young son had come home from school and told his mother, "Now I get it. Science is just like spelling. You just need to memorize it, and it doesn't make any sense." This is such a tragedy. Science taught is the dogma of scientists. Today in the United States, so many people who don't believe in vaccines think that, well, what science thinks about vaccines is just what scientists believe. I could believe whatever I want because scientists may be wrong and they don't have any reason to think what they're thinking. They're just making this up. They have no idea how science works. The same thing about climate change. So how adults have misdefined what science education means to students, I'll give you an example. This is true. My field is cell biology. As I've implied, the cell as the basic of life is the most amazing thing in the universe. The self-replicating chemical system. We can't understand how that simplest cell does it still, but it's just incredible that this is possible. In school, students should come in contact with real cells and learn how amazing they are, as well as how they work in general. They must take in food, they must put out waste, they must use energy, things like that. But instead, we try to fill their heads with all the names of their parts and their processes. And this is what we do to 12-year-olds in the United States. We have them read a textbook which talks about cells. And here's a sentence. Running through the cell is a network of flat channels called the endoplasmic reticulum. And that word is in bold because they're supposed to know what the endoplasmic reticulum is. This organelle manufactures, sorts, stores, and transports materials. Okay. And then in the same textbook, the chapter self-test says, write a sentence that uses the term endoplasmic reticulum correctly. And this is the only sentence they've had because there's not enough room to explain anything, and it's too complicated anyway for what they know. The next sentence was about the Golgi apparatus. That was another word they had to memorize. But this is meaningless education. It's not education. The critical part of the problem is much easier to test for science words than for science understanding and abilities, and bad tests force a trivialization of science education and drive most students away from science. And they have no reason not to think that this is just the dogma of scientists. They have no idea how science works. So a basic principle is that at all levels, teachers should make students struggle with a problem before they're told the answer. And research shows that this is a powerful way to generate deeper learning. Now, as I'll point out, this is true at college as well as at lower levels. Here's an example of what the first year school students can do in San Francisco. This was the actual part of the curriculum at one point. They put, at the right time of year when there's seeds on the ground, they put on clean white socks and walk around the schoolyard. In class, they come back, take off the socks, pick off with a forceps all the little black specks and put each black speck in a different numbered square on a piece of paper. Then they have a look at each speck with a $3 plastic microscope and draw a picture. And then they argue about which are seeds and which are dirt, and most are dirt. And some young kid eventually says, well, I think maybe the regularly shaped specks are seeds, and the ones that are not regular are dirt. And eventually the teacher, if they're running the class right, gets the class to agree that's a reasonable possibility. And then the next day the teacher might would say, well, how can we test this idea? And again, she doesn't tell the kids what to do, but some kid will say, well, maybe we could put all the regularly spaced seeds in one pot and plant them, and then put all the other specks in another pot and plant them, and see if only the pot with regular spaced shaped seeds gives us plants. And then eventually the class agrees, let's try that experiment. So this really shows that even in the first year, 5-year-old kids could do scientifically based experiments. Of course, as you go older, you do more sophisticated things. We have wonderful curricula and so-called inquiry-based science education. This is a bunch of 12-year-olds in San Francisco working in noisy cooperative groups. The teachers are in the back of the room like a coach, trying to help the kids solve problems that are more difficult than the one about seeds, but still appropriate for their age. The vision is to have an education that includes solving hundreds of such challenges over the course of the 13 years of schooling that lead to high school graduation. Challenges that increase in difficulty as the children age. I think children who are prepared for life in this way would be great problem solvers in the workplace, do things and make that are needed, and therefore have jobs that contribute to society. But even more importantly, they will also be more rational human human beings, people who are able to make wise judgments for their family, their community, and their nation. This type of education precisely fits the needs for workforce skills that have been widely expressed by U.S. business and industry. Here's what those are. This is from a book where they interviewed all the people who trying to hire people in their corporations. They want workers who have a high capacity for abstract conceptual thinking, the ability to apply that capacity for abstract thought to complex real-world problems, including problems that involve the use of scientific and technical knowledge that are non-standard, full of ambiguities, and have more than one right answer. I think this having more than one right answer is critical to learning how to think. The capacity to function effectively in an environment in which communication skills are vital in work groups. And now those students that you just saw working together, they're learning how to do this as part of their science lessons. So in the U.S., there's now a special opportunity. We have national voluntary standards in reading, math, and science. They all require major changes in how we teach. Students are to use information to sort, analyze, and critique it, to make and defend arguments, to solve problems and incubate ideas, or not to just memorize and spit back information like life is not a quiz show. It involves the ability to analyze and make sense of the world. Now, when I was at the academy working 12 years, I worked very hard on science education using the academy. And many of my colleagues said this was not our job. Science education before college was all about, somebody else, teachers, unions, textbook writers, you name it, anybody but college professors. But that's exactly the opposite of what I finally concluded. The rate limiting step in making science education much more effective at every level depends on changing the college level. Here's a simple diagram from another academy report that's showing how the poor teachers at lower levels are surrounded by all kinds of different effects, but we must remember that they learned what science education was by the science course they took in college, taught by the faculties of arts and sciences, by the scientists who are teaching them biology 1, chemistry 1, physics 1, or environmental science. An important barrier to progress is the traditional lecture format, which allows a single professor to process many hundreds of students through an introductory science class, now using a set of PowerPoint slides, and just like I'm doing right now. So I feel a little guilty. I'm giving a bad example of education, but I don't know what else I could do from this distance. Can we create much better alternatives without a great increase in cost? And at least in the United States, there's been great progress made. Here's the University of Minnesota, which has created many classrooms, a whole building full of, I think, 16 or 17 classrooms that each have 22 tables, each with 9 chairs, 2 computers, and an overhead screen, where kids are working just like those 12-year-olds in San Francisco, and they're teaching, for example, all their biology 1 courses this way. The woman who I put a star on the back, it's Robin Wright, who's one of the professors teaching this course. This is a picture of the active learning in college biology class. To do this, the University of Minnesota has to run their 3,000 biology students through many different individual batches. They have something like 200 students at a time in a room, so they have to run the course 15 times with different professors, but it's worth it. It's a completely different way of learning biology. So this is an important report that the Academy put out reaching students what research says about effective instruction in undergraduate science and engineering. It's a free PDF download at this web address. It's designed for college science professors, and it gives many examples of how to change your classroom, even in large regular lecture rooms. You don't need to have special rooms to make your education much more effective, as determined by scientifically based education research, seeing what happens when you incorporate active learning into science classes. This is a picture from that book I just showed. This is students working in groups using a computer simulation to investigate the properties of gases in general chemistry class. You see they're still sitting in a regular classroom, but they work and discuss with each other in that class during the lecture time. The lecture is interrupted every 15 minutes or so to give problems for them to work on. And this doesn't require computers. The so-called think, pair, share, where you, or write, pair, share, where you give them a question, write it down, write your own answer, share it with your neighbor, and then report back. All that kind of more active learning has been shown to be much more effective through research. Here's an example of meta-analysis that, from a group of science educators that showed that, that even mild forms of active learning have major effects on increasing learning. So I just want to focus near the end on 4 increasingly ambitious goals for science education, where we have still a huge amount of work to do, but I think it's very exciting and very important that we do it. So what are the goals of science education? I'm going to list 4. The first goal is to provide all adults with a general sense of what scientists have discovered about the world. For example, the vast expanse of the universe, all life is made of cells, the dangers of greenhouse gas accumulation in the atmosphere, so on. These important things that they should know in order to appreciate who they are and where they are, and how to deal with the world around them. So this has long been a standard view of the purpose of science education, and often it's just the only view. Adults need to know the facts that scientists have discovered. Well, there's too many of them. As you saw in that endoplasmic reticulum example, it gets drilled down to knowing complete trivia, which are meaningless to kids trying to learn them. So we have to do this better. We have to focus on only the most important concepts. We can't include all these facts. But the second view, which is, the view that motivated the kind of science education that I showed you with 12-year-olds or 5-year-olds, provide all adults with an ability to investigate scientific problems as scientists do, using logic, experiment, and evidence. That is how to investigate, why some things float and some things sink, and what does that mean, or why do seeds stick to socks, and which ones are seeds and which are dirt? All those kinds of things. All right, but now I think especially in the last year of shocking, depressing, irrational behavior by both political leaders and our citizens. After all, political leaders couldn't get away with nonsense if the citizens knew more. and they couldn't be fooled. So we need to provide all adults with an understanding of how the scientific enterprise works and why they should therefore trust the consensus judgments of science on issues like smoking, vaccination, and climate change. What scientists believe is not the dogma of scientists. It comes out of science as a way of knowing, a very special way of the community producing knowledge that no one person can make up. And that's, science is just an amazing human invention, and it's yielded so much more than the people who started it in the 17th century could have imagined. But most adults don't really understand how that knowledge from science arises. So we need a much stronger focus on this goal. We need experiments of how best to do this, and we need to share this understanding of how to do it across the world. And the fourth most ambitious goal, and probably the most difficult, is to provide all adults with the habit of solving their everyday problems as scientists do, using logic, experiment, and evidence. Being scientific thinkers on non-scientific problems, which are most of the problems they face in their lives. And I've been working on this. The education experts say that this will require a transference from what is learned in science class to general habits of mind. Not easy, difficult to do. How best can we do this? I'm just going to give you one of many ideas about that I've been struggling with and trying to do experiments with. Can a curriculum that includes reading an actual scientific paper be effective in empowering students with an understanding of the scientific enterprise? This could be done in high school, it could be done in college, for sure. So with this idea in mind, Science Magazine, when I was there years ago, started to develop a special free website for students and teachers. And here's the website. It's called Science in the Classroom. It contains carefully selected papers from Science Magazine. They're all short. We had a 5-page limit. And they were very specially selected to be understandable and relevant to students in high school or college. And they've been carefully annotated to make them much easier to read. That is, they have a glossary, a scroll over a glossary. The figures are explained and many other features that help with understanding of the paper and enable them to be taught more effectively. But how to use these papers to really efficiently allow students to grasp the nature of the scientific enterprise, how science generates knowledge that's reliable. That still needs much more work and experiment. So most broadly, we need to use knowledge of what increases student learning based on scientifically attained evidence to create a continuously improving education system at all levels. And this will require a much more focused and effective system of education research. This is something we also worked on at the Academy. Here was our first attempt to harvest what we know from psychology in academic studies. It's called How People Learn Brain-Mind Experience in School. It was published in 2000. It's the most widely downloaded PDF from the Academy website. A very successful report that was published when I was there. We also struggled with how to best to do education research. That was a report called the Strategic Education and Research Partnership in 2003. That led to this nonprofit organization that's still going called the SERP Institute to bridge research, practice, and design in education. There's the website if you're interested. The staff officer at the Academy, Suzanne Donovan, who was running that report on SERP, left the Academy to to be the executive director of this very successful enterprise with lots of resources, free resources on the website. I don't have time to talk about it here. So just want to end with the great importance of spreading scientific values with my favorite quote. This is from a book written by Jacob Bronowski, who's a physicist. It's called Science and Human Values, published in 1956, still in print. He was inspired to write this when he flew over the atomic bomb sites, Hiroshima and Nagasaki in Japan. He was a young man in the British Army, and he saw that damage, and it made him wonder whether science was bad or good for the world. He spent the next 10 years thinking and reading and writing this book. And of course, he concluded that on balance, science was enormously good for the world. He concluded that the Society of Scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. From these basic conditions which form the prime values, there follows step-by-step a range of values: dissent, freedom of thought and speech, justice, honor, human dignity, and self-respect. Science has humanized our values. Men and women have asked for freedom, justice, and respect, precisely as the scientific spirit has spread among them. And this is my last slide, very relevant to be talking across international borders. Science knows no country. Knowledge belongs to humanity. It's the torch that illuminates the world. Thank you.
I want to thank you, Doctor. Thank you for the lecture. May we start with the questions?
I stopped screen sharing. I see I've been off the screen. Sorry.
Just a little.
So my
May we start with the questions, Doc? See, let's start with the first one. Caio Magalhães asks you, Dr. Alberts, in your opinion, what was the greatest discovery regarding cell functioning?
Well, gosh, it depends on at what level. The 19th century, the discovery of heredity was an amazing piece of scientific discovery. I teach a course called, to graduate students, I still teach it once a year, called, sorry, I got to turn off my alarm, called Classical Papers in Biology. And we read the original paper by Mendel, I think 1863, it was just, breeding peas and figuring out that there was a logical, system for creating heredity. It was way ahead of his time, and it was just ignored and rediscovered only in 1900. And that was partly because during that whole time in the 19th century, really brilliant scientists have been looking at cells carefully with a microscope and making a really amazing discoveries, many of which are worthwhile looking at now. Let's see if we can explain them in molecular terms. There's a famous textbook of cell biology called The Cell in Development Heredity, published by Wilson, E.B. Wilson, American cell biologist. The first edition was 1898, and the third edition was 1925. The third edition is well worth reading still because of all the things that were discovered. But the the discovery that chromosomes were carrying heredity was really a wonderful discovery, made in a very interesting way, and really showing how science works. So I think that would be one of my favorite discoveries, the discovery that chromosomes carry heredity and Mendel's laws which really led us to the ability to really understand heredity in great detail.
Thanks. Let's go to the next question. Mayara Felix asked you, Dr. Albert, what are your expectations for molecular biology in the coming years? Thank you.
Well, I hope all of you are going to make wonderful discoveries. So, in our last edition of our textbook, we had a little section at the end, the 6th edition, what we don't know, important things we don't know. And that was a lot of fun for the authors. There's so much that we don't know that need young people to work out. I hope, let me just give you one example of something that I think would be critically important, both for understanding and for human health. So when cells, I used to teach medical students about cancer, and I would tell them that the most amazing thing about cancer is that we didn't all die of it before we were born. Because we have billions of cells, and if any one goes wrong and doesn't do the right thing, then it will kill us. It could kill us. If any cell is selfish and grows at the expense of the other cells, that leads to cancer and kills us. And an amazing thing is that during evolution, we had to evolve all these fail-safe mechanisms so that you could have a cellular organism. It took a billion years or billions of years to develop these safeguards, so that, and one of the safeguards is when cells have a detection system, when anything is going wrong and they're not behaving right, they kill themselves. Well, cancer cells, eventually, with accumulating 10 or so mutations, they're able to escape from that killing process. The killing process is called apoptosis. So if we understood the signaling network that tells the cell it should kill itself, if we really understood how it worked in molecular detail, and could therefore in every cancer cell see how that system has been changed, I think we could kill most cancer cells without harming normal cells using that knowledge. So, one of the things that I think would be a wonderful focus for a group of people with different talents. You need people with computer network understanding. You need people with biochemical understanding. You need all kinds of talents to work together to try to really decipher that network in absolute detail. And that will be a major discovery. I'm just talking about things that have relevance to the medicine, and the other thing that happens is that's really a tragedy for humanity is all these Alzheimer's and other degenerative brain diseases in old people, which is just terrible burden for humanity. We'll all be taking care of our old relatives pretty soon. So we have to figure out how that happens. And there's a huge amount of work going in and trying to prevent Alzheimer's disease, but we should really be focusing on some of the basic understandings that are going to be needed to before we can cure that disease or prevent that disease. I think the one idea I trying to suggest to one of our cell biology journals is to have groups of textbook writers who have had the chance to been forced to think about this, prepare some regular articles. Well, here's some problem that needs work, and here's the kind of things that need to be done, and do that over and over again to help guide younger people to really important problems. It's too easy to work on. It's so easy just to measure things. You don't want to waste your scientific life just doing something. You want to make a difference. And maybe some of us older folks could help by encouraging people to work on issues that nobody's working on, that really will in the long run be important. The things that are immediately obvious, like we need some drug that will stop Alzheimer's disease, everybody's working on that. But we have to go back much further and say, what kind of understandings would help us, and work on those.
Okay, the next question is from Maria Zanella. Dr. Albert, how it is to be a science in the USA?
Well, I think it's at least better than being a scientist in Brazil right now. I've heard about all the cuts in your budget. It's terrible. So I've written paper. Me and my colleagues founded a little organization called Rescuing Biomedical Research. Harold Varmus, Shirley Tillman, I, and a few others. And we wrote a paper in 2014 about some of the problems. And we focus on young people. We need to work to make it much easier for young people to work on difficult new problems, because it's very much easier to get funded now to work on something obvious than to work on something non-obvious and hard. So when I talk to young people starting their own independent research careers, so many of them feel they can't really work on what they think is an important mystery, because it's too unlikely to get money from the government. So we're trying to create a system. I think right now the European Union is doing better than the United States, because they have the European Research Council, ERC, that has something called starting grants for young scientists, and they're looking for new ideas, risky ideas, and not on what you've already done and keep doing it. So we want to create a system like that in the United States, where we have many more opportunities for young people to get funded for new ideas that are risky than we now have now. We have some grants that are like that, the New Innovator Awards, but we need many more of them. So, I think it's hard for scientists in the United States right now to work on the kind of problems that I think many of them should be working on, but we're hoping it's going to get better very soon. There's going to be a lot more money for science very soon. So important to me is to try to use that money well. My friend Eric Lander is the new head of science. And he's a science advisor to the president. He's a member of the cabinet. He understands these things very well. So we have a chance to I think, in the new administration to make the system better for young people than it is now.
Thank you. And let's go to the next question. Jose Roberto asked you, Dr. Alberts, what were your biggest challenges as a president of the National Academy of Science of the U.S.?
Well, one of the things we really tried to do was create an international scientific enterprise where the scientists were all working together and be much more effective if, for example, if all the academies of the world worked together than if we're all doing things alone. So in 1993, we had the first ever meeting of all the academies of the world. This was organized by the Indian Academy of Scientists. It was the first time I ever went to India. And that meeting was so successful. We were working together to work on what does science say about world population issues? What should we do about the exploding population and how to deal with that? That we founded something called the Inter-Academy Panel, which still exists. It's now called the IAP, Inter-Academy Partnership. And we founded a organization that part of that was to do studies like our academy did for our country, to do similar studies for the world using all the academies together. And I worked very hard on that for years. I think that kind of effort needs much more emphasis. And part of the trouble with the work that we did back then was that the older academies are too old. I mean, most of the people, first of all, they're not on social media, so we don't exist for most of you. But more importantly, older scientists are very busy with things they already are or committed to, and most of them have not had enough time, didn't have enough time and energy to put into these kind of international sharing events. And more recently, the German Academy in the year 2000 invented something called the Young Academy. I never thought of this. It's a wonderful invention. There are now 40 nations with young academies. What is a young academy? Young academy are 35 to 40-year-old scientists and engineers who are brought together, about 100 of them. They're in the Young Academy for 5 years. The German model has been copied first by the Netherlands, and now by 40 different countries. So you might have 20 new members coming in every year and 20 members going out. These young academies, I think, are more effective than the old academies in working across international boundaries because, first of all, they're all over the internet and they're all over social media. And second of all, young people don't have to sleep so much. They have much more energy. And so I'm a big fan of increasing the force of international science through harnessing the energy of younger people. So the young academies are 35 to 40-year-olds. We just starting one in the United States. I couldn't get my successor interested, but now Marcia McNutt, the new president of the academy, we have a young academy. It's called New Voices, but there's a network of young academies, and I'm talking in the right place because South America is very deficient in young academies. There's hardly any young academies in South America. I don't know why. I've been talking to people in Brazil who want to start one. I've been talking to people in Chile and Argentina. But so far, the young academy movement has not taken off in South America. And if you look at the map of young academies, which I could have shown on the slide, South America is almost empty. And of course, even the United States was absent for far too long, so I shouldn't be very critical. But anyway, I think the answer to my question is we need much more force from science by bringing together scientists from around the world. Academy-like institutions can in principle be very effective in doing this. The older academy networks work very hard on inquiry-based science education. They've done a lot, actually. France was one of the leaders of that effort. They have a very important academy effort there to get the kind of science that I was talking about spread throughout France. And they also are working in many different countries. So the French and the Chileans were actually very active in that too. And so, that's a start. But we need much more. Like on the issue that I've been talking about, figuring out how to teach all children and all college students, make them really understand where scientific knowledge comes from. How do we know that vaccines are safe? Why should you listen to the scientists at all? Because, are they more than just some belief system? I mean, I know from a fact that when we do interviews of Americans, so many people just think they could pay attention to science or not pay attention to science because science is what scientists believe. It has no special, they don't see that it has any special value, that it comes from a different place than what other people believe, right? I mean, in America, we used to cure diseases, try to cure diseases by burning witches, right? It didn't work. But now we're back to that kind of crazy magical thinking, microchips in the vaccine are from Bill Gates and they're going to control your life. I mean, this is crazy.
Okay, let's go to the next question. Fee Santos asked, "Hi, Dr. Albert. Do you think that programming skills will be necessary in the future for biochemistry analysis, or that skill is already necessary now?"
Well, I think you certainly need computers for a lot of things. Programming skills, I think, will be important, but no one person can do everything. I think what's really important is to be open to collaboration with people who have skills that you don't have. There's no way that any one person could do everything that's needed. So, sure, programming skills will be very important if you could get some of them. But a lot of the problems, like emergent properties that I'm talking about, are requiring very sophisticated computer science. And so it'd be great to focus on that, but you can't expect to do that and also be a biochemist and also be, an expert in light microscopy, another major breakthrough in technology. There's so many important technologies that need to be brought to bear on many different, whatever you're working on, whatever mystery you're trying to solve, you should be able to bring all those tools that are available to bear on that mystery, but you're not going to be able to do it yourself. So collaboration is going to be very important. As I said, strategy is everything. I want to solve this problem, okay? So I want to solve the mystery of apoptosis, okay? Then I need to really do a lot of reading and a lot of thinking and come up with a clever way of making progress that will at least solve part of the problem. And then I have to decide, which of those skills can I get, and which of the skills do I need to work with others to get. So, for example, I tell graduate students, if they do their PhD in biochemistry, say, and they learn how to do the kind of work that I did, well, then before they do their postdoctoral research, that's the standard before you become an independent researcher, is to get a PhD degree, then work for 3 or 5 years in another lab as a so-called postdoctoral researcher. I tell our graduate students after they're getting their PhD, the best thing to do is to think about a mystery you want to solve 10 years from now, and then try to think about what kind of skills you're going to need to solve those, and then do a postdoc to get you that skill. Don't work on the same thing you did already. The tendency for graduate students is to go and do a postdoc with the most famous person in the field they've already worked in, but then they're not going to learn that much. You want to make yourself unique. You want to know 2 different things that most other people don't do together. And there'll always be people doing any one of the ones, but if you have two different skills, that's going to make it more likely you can make an important contribution. So, there's a huge opportunity here to solve new problems, and I hope that many of the people listening to this will be major contributors.
Okay, the next question is from Henrique Almeida. Dr. Alberts, in your point of view, what should be the role of a chemist in the next developments of the cell biology?
Well, it depends on what kind of chemist you're talking about. There's computational chemistry, that's all the emergent property stuff. There's a huge amount of technology development that's dependent on chemistry. What's really been impressive in my career is how the tools that we have for attacking problems have become much more powerful and much more creative. And so you have a choice when you're designing a strategy to solve your problem of using so many different tools. And some of those tools have been developed by chemists, the ability to label any individual protein in a cell and follow it. There are huge numbers of clever things that are still being developed. One of the fields I write about, I used to work on it a little bit, not very effectively, but my chapter on chromosomes has a big section on chromatin, that is, and on heterochromatin. The way of packaging DNA in histones and nucleosomes so that you have an inherited pattern of turning off genes. This is very important for our existence as multicellular organisms, to keep genes off in a heritable form and then turn them on when they're needed. And that system goes wrong in cancer often. Anyway, so we need to understand what is heterochromatin biochemically, and how does it actually work, and what goes wrong with it in cancer and all that stuff. Well, we need new tools. It's a complicated structure. We need new tools. Chemists are working to make new tools so that you could, for example, construct a pure system of DNA with nucleosomes on them that are specifically marked with different covalent methylation or acetylation patterns. Those patterns form some kind of a histone code that we don't understand. And to form heterochromatin, you need to have this lysine of this histone methylated, and this other lysine not methylated, and all those kind of things which require chemistry to reconstruct substrates for studying them. An important, totally unrealized and not actually being researched very well because it's hard. One goal would be to reconstruct with purified components in a test tube the structure of heterochromatin. There are different kinds of heterochromatin. Take one kind of heterochromatin and try to reconstruct it exactly so we can understand it. Before we do that, we're not going to be able to understand how it works and how it goes wrong in cancer and what we might do about it. So that's just one of many examples where we need clever chemists. Obviously, chemistry is heavily involved in drug design. But as cell biologist, I think I'm not really involved in trying to cure diseases myself. I think more about the ways in which chemists have devised little clever tricks to help us do better experiments.
Thanks, Dr. Albert. We have 3 more questions. Is there any problem to you to continue with us a little more?
Yes, sure.
You may stay a little because we are almost on time.
Okay, well, keep. You have another good question?
Yes, we have 3 more.
All right, three
Okay,
more. Three more. Okay.
Isabella Rocha asked, do you think omics science could help in the future to understand how the human body works and how it responds to diseases? And maybe could it be useful to find a cure for these diseases?
Yeah, of course. Every kind of science has a role in all of this. Omics science. Well, I'm critical of some of the projects that we're now spending huge amounts of money on, like Project Encode, where they just keep measuring things, right? We're measuring huge amounts of information. It's easy to collect data. Much harder is to collect data in a clever way that tells you something interesting. And I'm critical of a lot of modern omics science, where you have these huge labs turning out information about, which gene is expressed in which cell and at what levels. I mean, we had a big controversy. We write about this in our textbook now. In 2012, this Project Encode project had a lot of publicity. They could measure how much RNA was in a cell down to less than 1 molecule per cell. And with this very sensitive technology, they discovered that you could find RNA sequences that matched 80% of the human genome, and they concluded that 80% of the human genome was functional, and that there were all these RNA genes that were making only RNA and not protein. Well, the fact is, as a physical chemist, that any process is going to have errors. I mean, everything in biology, cells are amazing for many reasons, but one of the reasons is that, they can't avoid mistakes. We replicate DNA with error, about 1 error in 10 billion base pairs or something like that. But it requires all kinds of correction mechanisms to get to there, because we can't make it that accurate, because the base pairing has to obey physical chemical laws. And so with imperfect base pairing, the cell nevertheless had to devise a system to be very effective in replicating DNA and repairing DNA. So all those constraints are really critical. And so what we believe is that all those things that those omics people were measuring were just random errors of making a little bit of RNA. So, we have to still fight the misconception as textbook writers that there's no junk DNA. Well, we know there's junk DNA. There's lots of evidence. Only about 10% of our DNA sequence has function. So, spending all that money measuring things down to less than 1 molecule per cell didn't make much sense to me. But omics done well is important.
The next question is also from Isabella Rocha. She asked, do you think the new generation of scientists should reach the changes you talked about in science education? Why it is so hard for scientists to communicate with people?
Well, first of all, it's true that when you understand something, it's very hard to teach it. If you understand it too well, I think my best teaching was when I was teaching something I had to learn just before the lecture, because then I understood what I had to say to get people to understand it. But when you understand things really well, you tend to assume everybody else has the background you have, and you make yourself completely unintelligible. People can't understand what you're saying. So I think scientists learn how to communicate by by teaching more effectively. But let's see, what was the first part of the question? Oh yeah, we have a big role to play in science education. That's what I'm saying. Well, let me just talk about my grandchildren. I have 7 grandchildren. I want them all to be science teachers. But they're not going to be. But some of them are going to be science guys. Because I think one of the most important things we could do as scientifically trained people is to teach others. And teaching high school science is critical if you teach it well. High school science teachers could make adults, understand why they should be vaccinated, or whatever the next new thing is, or why they should pay attention to climate change. Make them appreciate what science is. And what amazing, one of my friends who wrote, science is really, what science has developed is really a miracle. You wouldn't have expected it to be so effective. And yet, of course, so many people don't understand or appreciate science. So at any rate, I think scientifically trained people are valuable in many professions, not just doing scientific research, but science journalism, science in politics. We have a program in the United States that sends young scientists to work with politicians in their staff. That's critical, important. I think to spread science, we need to spread scientists, which means providing career opportunities for scientifically trained people to do many different things in many different places.
Now is our last question. William Piero Salgado asked, with all this new technology being developed, like the synchrotron concatenation, etc., do you feel that more than ever, scientists from different fields should talk and communicate with each other?
Yes, of course. I also think that our Zoom, the pandemic has been useful in creating a new, what we call Zoom culture. Everybody knows how to use this new tool to work together. And we're doing some experiments in science education, actually, where we're bringing people together across the world to try to get them to collaborate. 10 people who never would speak to each other otherwise are spending 2 hours every few weeks trying to solve a problem together. And that could be done, should be done, and is being done in science too. And so the fact that we don't have to travel anymore to really try to create a cooperative thinking endeavor and solve problems, I think is a great opportunity for science in exactly the way that was described. We could get people to spend a little time thinking about other kinds of problems that they otherwise would never think about, and to interact with people who, I'm a trustee of the Gordon and Betty Moore Foundation, which started a new program trying to understand symbiosis in the ocean, how coral reefs survive, for example, with algae. And they've created a Zoom-based culture of 15 scientists who work on very different things and have very different skills. I think that's working really well. And, it would have been very difficult. I mean, you could always have telephone conversations with 15 people, but you never know when anybody's listening. So it's so much more effective in ways we couldn't imagine to be able to look at people's faces to make sure they're still paying attention. So those kind of meetings, I think, could be exploited tremendously in science and greatly improve science. So thank you for that question. It's a very good question.
Well, Dr. Bruce, I thank you again for the lecture. It was an honor having you here in our event.
Well, it's my pleasure. I've been in Campinas once, and I hope to be back someday soon.
We hope that too. And I also would like to thank, on behalf of all the Unicamp's Chemistry Week Organizing Committee, our sponsors. And last but not the least, I would like to call Maria to do the Introdução à Bioquímica book giveaway. Dr. Albert, if you have anything else you need to do, there's no problem if you need to leave, okay?
No, I could get. You want me to leave? I could leave. Okay.
No, please. Only if you need to.
Okay. Okay. All right. Go ahead. I'll wait.
Eu vou sortear, aqui estão todos os inscritos na palestra. E sortear um número de 2 a 1530, que são o número de inscritos. Número 367. Então, a sorteada foi Márcia Ferreira da Silva. A comissão vai entrar em contato com você para pegar seus dados para você receber o prêmio. Obrigada. And thank you, Dr. Bruce.
Thank you again, Dr. Bruce. And good afternoon to you all. Bye.
Bye-bye.